Today, we'll learn together the basics of plant cells, and while there is plenty of good material in YouTube about cells, I don't want to make a lesser video than what is already available. In fact, I'll be using clips from Harvard and Nucleus Medical Media cell animations to reinforce the analogy I'm using when teaching this topic inside the classroom. My primary focus here is for you to have a good grasp of plant cell structure and function via this simple analogy. This is Professor Bryan Yves Araneta. Let's start. Think of cells as protein factories, and this is the reason why the central dogma of cell biology is written this way. Proteins are the ultimate goal of all cells to ensure that they exist and behave like they do. You will learn that in future discussions, but for now, I hope that a noodle factory will make a good analogy. Even if you have all the necessary materials and equipment and manpower, you cannot manufacture a single noodle without the recipe. In short, you need information. In the case of cells, all information to produce all the proteins needed by the plant cell is encoded in the DNA that is found in the nucleus. It is kept safe there and covered by the nuclear membrane studded with characteristic pores that allow exchange of materials between the opposite environments. Normally, a factory doesn't produce all the products written in the recipe library, right? Production usually focuses only on what is in demand at the moment. Thus, in the recipe book, only information about one particular noodle is used at a time. It contains all the information about how to process the product, including the specifications of the equipment to be used. In cells, these pieces of information coding for a single protein can be found in segments in the DNA called genes. And to extract them, they must be transcribed into shorter messenger RNA segments. RNA synthesis happens in the nucleolus. That's why I compare it to a photocopying machine, because it copies pieces of information from a larger DNA to a smaller RNA molecule. You might wonder, why do we need RNA if there is already information in the DNA? It is because DNA cannot leave the nucleus and protein synthesis occurs outside the nucleus, in the cytoplasm. The nucleolus is also the site where ribosomes are assembled. When ribosomes, together with the RNAs, are released from the nucleus, it will start making proteins by joining together amino acids in the cytoplasm. Ribosomes are thus the actual sites where proteins are being synthesized from their amino acid subunits. In our analogy, these are the noodle machines that now uses the specified ingredients and produce the product. Depending on their location, some are referred to as free ribosomes if they are just floating or freely moving around in the cytoplasm. Otherwise, they are called bound ribosomes if attached to structures called the endoplasmic reticulum. These are then referred to as rough endoplasmic reticulum because they look that way with ribosomes on their surfaces. In this analogy, I represent the endoplasmic reticulum as a sort of a conveyor belt, which moves the newly produced noodles to where additional processing procedures will be done. In cells, the endoplasmic reticulum, or the ER, is responsible for transporting the newly formed proteins to the sorting, packaging, and labeling sections of the cell. The bound ribosomes drop the raw proteins into the ER for transport via membrane-bound vesicles. That is why it is a general rule that proteins produced by bound ribosomes are destined to be transported out of the cell, while proteins produced by free ribosomes are only for localized needs. So remember that the rough ER plays an important role in protein trafficking via transport vesicles. There are, however, ERs that has no attached ribosomes, thus referred to as smooth endoplasmic reticulum. If present, they are known to store lipids and essential ions or minerals needed in the various metabolic activities in the plant. It is clear that the noodles coming from the machines are not yet sorted, packaged, and labeled. And some additional stuff like flavorings, oil, and vegetable bits, for example, are yet to be included. In cells, the raw proteins produced by the ribosomes are also to be modified. They also need to be sorted, tagged with specific molecules, and additional segments like carbohydrates and fats can be added to make the proteins functional. That is done in the Golgi apparatus. In the school animations here, the proteins from the rough ER are transferred by the vesicles to the Golgi apparatus. I greatly recommend you watch these full videos, and although those are animal cells, the rendition of the relevant organelles share the same conceptual basis for plant cells. In our analogy, of course, the Golgi apparatus is represented by the sorting, packaging, and labeling sections of the factory. Then, the packed and labeled product is now ready for transport to the market or to where they are needed. Transport vesicles budding off from the Golgi may fuse with the cell membrane to release it outside the cells, so it can be transported to the different areas in the plant. Otherwise, these products are just transported within the cell. The cell membrane is a lipid bilayer and is associated with the Golgi apparatus, the endoplasmic reticulum, and the nuclear membrane. That is the reason why all these structures are membrane-bound and collectively referred to as the endomembrane system, just like a network of customers and routes for the delivery truck. So far, we have looked at the primary structures that carry out the primary functions of the factory. There are structures that have support functions which are essential for the overall operation and well-being of the factory. Obviously, plants need the mitochondrion, where sugar molecules are being burned for energy in the form of ATP via a process called aerobic cellular respiration. It's not hard to imagine a factory without electricity. Aside from the mitochondria, plant cells also have chloroplasts that harness solar energy to produce energy molecules like NADPH and ATP. But these are mainly used for fixing carbon dioxide into sugar molecules. Many green tech farms or factories nowadays do not solely rely on mainstream sources of electricity by using supplemental solar power grids. Plant cells are like that too. They are self-sustaining because they can produce their own food via photosynthesis using solar energy in chloroplast. And this food can be burned via cellular respiration in the mitochondria to produce energy when needed. Vacuoles are like water and salt reservoirs in cells, and is represented by a water tower here. Because the internal environment of cells are made primarily of water, there is frequent change in its osmotic properties that water or salts are required to balance it. Plants noticeably have a large central vacuole, which is also indicative of the greater need of plants to store water for photosynthesis. The cytoplasm is the semi-solid matrix that covers the entire internal environment of plant cells. That is a no-brainer. And then we have the cytoskeleton that provides the framework that supports the cell. Just like the framework of the noodle factory, the steel bars, the concrete, anything that maintains the structural integrity of the unit. These are made up of proteins and categorized into microtubules, microfilaments, and intermediate filaments. They are dynamic frameworks because they can assemble or disassemble when needed to adapt to the needs or shape of the cell. They also guide the movement of transport vesicles. I really love that animation where motor proteins move vesicles along protein filaments. And finally, we have cellulose cell walls in plants, which acts as a fence or a wall or a protective covering that ensures that the manufacturing process inside is kept safe from external disturbance, both chemical or physical. In some illustrations, there are pores in the cell wall called the plasmodesmata. This allows communication between adjacent plant cells and also acts as a passageway for materials. Alright, what else have we missed? Lysosomes, yes. Some experts believe that lysosomes are not plant cell organelles, but some also report the existence of lysosome-like vesicles in plants. Just for the sake of covering everything, lysosomes are modified transport vesicles that contain digestive enzymes. These enzymes are involved in the intracellular digestion of unused materials inside cells. So this is much like the recycling plant in the factory. There are also similar vesicle-derived microbodies called peroxisomes or glyoxysomes, which are involved in lipid metabolism and getting rid of toxic substances inside plant cells. We're done with the eukaryotic plant cell. Let's now look at the simple configuration found in blue-green algae, the plant representative of prokaryotic cells. This is a plant bacteria. That sounds ridiculous, but anyway, as a prokaryote, it only has a simple internal organization. Let's start with the cell wall, which is made up of peptidoglycan, a combination of proteins and carbohydrates common in bacteria. Take note that cellulose is the primary material found in cell walls of higher plants. Here, there is an additional layer consisting of mucoid sheath, capsule, and slime coat. These are sticky polysaccharide material mostly for adhesion or allows gliding movements in solid surfaces. It also has the cell membrane, and inside it is a network of folded membranes called the thylakoids. These contain enzymes and pigments that trap light energy and convert them into ATP. There's the cytoplasm, a semi-solid matrix that also contains enzymes for sugar production. Because prokaryotes are primitive organisms, the DNA is only localized in this particular region called the nucleoid, and not really enclosed in a membrane, just like the nucleus of eukaryotic plant cells. Since prokaryotic cells also produce proteins, ribosomes must be present too. But of course, without the network of advanced structures like the ER and Golgi apparatus, there is little additional processing of protein products, or none at all. This is just a simple cell with simple needs. There is no mitochondria here, and the cell produces energy directly from the photoreactions happening in the thylakoid membranes. The cell membrane also contains enzymes for extracting energy from food molecules in the external environment. I think I'm done. At least I didn't leave the prokaryotic cells behind, and I hope that my noodle factory analogy for eukaryotic plant cells doesn't sound terrible. Thank you guys for watching. Subscribe to my channel, like this video, and share it too if that was helpful.